Okay, so I think we'll go ahead and get started. So welcome everyone to the Multicultural Alzheimer's Prevention Program, or MAPP Rounds. My name is Talia Robinson. I am a postdoctoral fellow in the Harvard Partners Consortium in Clinical Neuropsychology. And it is my pleasure to introduce Dr. Kacie Deters, who will be giving us a presentation entitled Exploring Genetic and Neuroimaging Risk Factors for Cognitive Aging in Black Individuals. Dr. Kacie Deters is a postdoctoral fellow at the University of California, San Diego in the Department of Neurosciences. Her research focus is on ethnic and racial disparities in cognitive aging and Alzheimer's disease in older adults, primarily from the Black community. Dr. Deters is very passionate about DEI and currently serves on the advisory board for the Resource Centers for Minority Aging Research. So we are very happy to have her presenting today. And some learning objectives that she will be covering include identifying disparities for risk factors associated with brain health specific to the Black community, understanding how genetic and neuroimaging risk factors impact brain health and cognition, and identifying gaps to address in order to minimize cultural biases in recruitment for clinical trials and cohort studies. So before she gets started, just as a reminder to please use the Q&A to write questions. So we will address questions at the end of the presentation. And this meeting is being recorded, and the recording will be available tomorrow on the MAPP YouTube channel. So without further ado, welcome Dr. Deters. We're happy to have you.
Thank you so much for the introduction. That was wonderful, Talia. And I'd also like to thank the organizers, Marie and Shaquille, for inviting me here to discuss some of my past and current research. So yeah, I'll be talking about genetic and neuroimaging risk factors for cognitive aging in Black individuals. I'm sure this is nothing new to this group, but Alzheimer's disease is the most common form of dementia. The two hallmark pathological features of Alzheimer's disease are extracellular amyloid plaques and intracellular tau tangles. Another key characteristic of Alzheimer's disease is progressive cognitive decline, as shown here in the y-axis. And over time, as shown on the x-axis, some cognitive decline is completely normal in normal aging. However, before overt dementia is present, individuals that might show brain pathology of these proteins have a stage called preclinical stage. Then as time progresses, when symptoms become present, you develop mild cognitive impairment and dementia. We can track the trajectory of this continuum of dementia stages from preclinical on using biomarkers. As shown here, and as I alluded to in the previous slide, protein deposition, amyloid and tau, begin decades before symptom onset. For example, if we look at the red and green line, which represent amyloid and tau respectively, we can see that abnormal levels are present in the brain before the MCI stage. Abnormal accumulation of these proteins leads to neurodegeneration, which can also be tracked using structural or volumetric MRI measures as represented by the blue line here. Finally, we can see cognitive impairment, which can be measured using an array of neuropsychological assessments, as represented by the purple line here. However, all these biomarker trajectories were identified in mainly non-Hispanic white participants, which presents a really big gap in our field regarding how do these trajectories look in non-white participants. So I want to discuss why research within the Black community is important. So Black Americans are disproportionately more likely to have cognitive impairment and dementia compared to white individuals, which is shown here. And some data from Kaiser Permanente in Northern California. We have incidence rate of dementia here on the y-axis and age on the x. African Americans are represented by this purple line, and white individuals are represented by this coral line here. And on average, Black individuals were twice as likely to present with dementia relative to white individuals. Yet whiteness continues to be centralized in research. For example, there's this recent study that shows individuals of European descent, which is shown here in the blue, make up more than 3/4 of the participants in genetic studies, as opposed to the African group, which is only represents 2%. So how can we properly assess risk factors, genetic and beyond, if we have such limited genetic ancestry or genetic information from the African group. And this is really important because the United States is becoming more diverse. This is US Census data for the past 20 years, starting 2000 and 2019. And for the first time, we're starting to see people who identify as white in the United States is declining in the past 20 years, while every other group is increasing in representation for the United States. So there's a real need to bring justice and equity to research practices for Black community, but also for other minoritized groups. So I want to talk a little bit about an example of why it's important to focus on identifying risk factors within non-white communities. So this plot here shows cognitive score, global cognition on the y-axis, and age, or you can think of this as time as well, on the x-axis. And we can see two key features. First, at baseline, which is here with this green box, we can see that Black individuals have a lower average cognitive score relative to white individuals. However, when we look at the slope of cognitive decline between the two groups, we see that there really isn't a difference in the rate of cognitive decline between the two groups. And while there's been conflicting studies to show this, on average, this appears to be the trend. So this highlights not only racial bias in neuropsychological exams, but also the need for longitudinal studies in Black cohorts, especially if for some reason, we continue to pursue research on disparities between Black and white individuals. So next I want to talk about genetic risk factors and how they are not the same in Black individuals. On chromosome 19, excuse me, on chromosome 19 lies two genes that confer risk for cognitive impairment. And the first that I'm going to talk about is one I'm sure we're all familiar with, which is apolipoprotein E, which contains E2, which is thought to be protective, E3, which is present in the general population, and E4, which confers higher cognitive risk for not only Alzheimer's disease, but also cognitive impairment over time. And you'll notice I left out the E2, E4 carriers, and that is because the risk of this genotype is unclear, considering the protective and risk nature. So you might think that having a higher frequency of E4 might equate to a higher incidence rate of Alzheimer's disease. However, that is the contrary of what some evidence shows. So for example, in the solid bars is the percent of E4 in cognitively normal, older, healthy controls. So as you can see, there's actually a higher E4 frequency in Black individuals relative to white, cognitively normal, healthy individuals. However, when we look at the incidence rate of Alzheimer's disease, which is shown in these pattern bars, we can see that the percent of E4 is almost the same between the two groups. So again, suggesting that although E4 has this higher risk of Alzheimer's disease, it may actually have a reduced risk in people who identify as Black. And there's some potential hypotheses, but I want to highlight this recent paper from the Vance group out of Miami that looked at ApoE4 expression in individuals of European and African descent, and actually showed that there was higher ApoE4 expression in individuals who were of European descent as opposed to African descent. Just upstream of ApoE is the TOM40 gene. Within this gene lies an intronic variant that codes for poly-T alleles. We'll just call it 523 for short. And as shown in this figure here, it typically is associated with repeats between 15 to 40 T alleles. And this genetic variant is associated with increased cognitive impairment and Alzheimer's disease risk. However, TOM40 is also in linkage disequilibrium, or meaning, these variants with ApoE and TOM40 are typically inherited together. So any risk conferred by TOM40 was originally thought to be due from ApoE4. However, it was discovered that the variation in this TOM40 523 variant actually differed by ethnic and racial background as a function of ApoE. And I'll give an example of what that means. So if we look at the TOM40 length for people who identify as non-Hispanic white and non-Hispanic Black in cohorts from Rush University, the number of poly-T repeats looks almost identical. It's almost as if there's no variation. However, when you look at the number of poly-T repeats on the APOE 3 background, we can start to see that it was associated with poly-T repeats less than 20 and greater than 30, as opposed to E4 carriers who had poly-T repeats between 20 and 30. So we're going to refer to these as short, long, and very long repeats. And when we look at if this pattern is the same in Black E3 or E4 carriers, we can see for Black E3 carriers, the number of poly-T repeats was similar in the fact that it was short and very long. However, if you look at Black E4 carriers, they have patterns of short, long, and very long poly-T repeats relative to the non-Hispanic white E4 carriers. So I was really interested in investigating what this variation might mean for cognitive decline. But before we continue, I'm sure you're asking, where did this variation in Black E4 carriers come from? So I want to remind you that this work is focusing on individuals who self-identify as Black in the United States. Excuse me. Because of forced migration and the atrocities that were inflicted on our ancestors, people who identify as Black in the United States are often genetically admixed between African and European ancestry. So I have an example here from a data set that shows African ancestry, which is in red, that exists on this continuum from 30% up to 80% in this cohort. And they have almost the exact inverse of European ancestry. So while I'm showing data from a past study, which is shown here from Allen and Roses et al., I want to talk about where this genetic diversity might come from, and that is looking at the Yoruban population, which is a ethnic group in Nigeria, and the poly-T repeats that they have is very similar to Black Americans in the sense that Yoruban E4 carriers have short and very long, and this little bit of long here between 20 and 25, while people of European descent have seem to be contributing this longer portion, having between 25 to 30 poly-T repeats to Black Americans. So I like to think of this APOE and Tom40 variation as a really great example of genetic admixture. And just before I get into data, I just want to give a brief introduction to what genetic admixture is. So if we have two populations from different regions, say from West Africa, which like the Yoruban population, Yoruba group, and we have a group from a European culture, most of our DNA is exactly the same. However, if there's a specific loci in which, for example, the A allele is present all the time in people from this West African group, but the G allele is present all the time in people from this European group, this is what we call an ancestry informative marker. And it's supposed to, imply ancestral information. So if we wanted to calculate global ancestry, after many generations, a person who identifies as Black in the United States or as biracial, such as myself, would have admixed DNA from both regions. And if we wanted to quantify the amount of African ancestry, which is the green, we would divide that by the total number of ancestry informative markers, multiply it by 100, and end up with a percentage. And so that's how I calculated global ancestry for these groups. This highlights another really important feature of genetic ancestry, and that it is reflective of geography, not race. If anything, again, focusing the fact that we're identifying variants from groups in specific regions, I am just trying to make a case that genetic ancestry has nothing to do with racial categorization. So this leads to the first study that I did in collaborations with Dr. Lisa Barnes at Rush University, and that was to look at APOE and Tom40 haplotypes with cognitive decline. For this study, I used Rush cohorts. They enroll at the age of 65 or older and without known dementia. And I used 3 studies: the Religious Order Study, the Rush Memory and Aging Project, and where the majority of participants came from was Minority Aging Research Study, or MARS, which collects data from Black individuals from Chicago and surrounding suburbs. They have a really rich, comprehensive neuropsychological battery of a number of different cognitive domains that are all averaged to create a global cognitive measure. So again, the most interesting thing to look at was this variation in Black E4 carriers. So I wanted to determine, is this associated with cognitive decline in E4 carriers? And before I get to the results, I just want to, for statistical purpose, we had to have some type of groupings. So I decided to look at this in the sense of number of short alleles. So an individual could be short, which is represented by the red line here in the graph, having one short allele, or having no short alleles, which is represented by the gold in the graph. So as you can see, being Black, E4, and having no short alleles, you had a significantly faster rate of cognitive decline relative to the Black E4 carriers. Where's my mouse? The Black E4 carriers who did have a short allele. When we looked at this in relation to the white E4 carriers, they are just one line, because as a reminder, there is no variation. They just are represented by long. So we see that baseline effects such that Black individuals have a lower cognitive score relative to white E4 carriers. However, if we look at the white E4 carriers to the Black E4 carriers who did not have a short, this was not significantly different. They had the same rate of cognitive decline over time, which the average was around 8 years. But when we compared white E4 carriers to the Black E4 carriers who did carry a short allele, not only did this group have significantly faster or slower rate of cognitive decline relative to the Black E4 not short carriers, but the Black E4 short carriers also had significantly slower rate of cognitive decline relative to the white E4 carriers. So this suggests a potentially protected group against the determinants of APOE4. So moving past genetics, I want to talk about some proteins. Postmortem neuropathological insults are also not the same in Black individuals. In sticking with data from the Rush cohorts, this is postmortem neuropath data looking at in Black participants and white participants. And pure AD is amyloid and tau. And we can see that people who postmortem brains that were diagnosed with Alzheimer's disease had less of this pure AD relative to white participants. So there's higher comorbidities, neuropathological comorbidities in Black participants relative to white. However, there has not been a lot of biomarker data research in Black individuals. So at this time, we really only have a good number of information on tau, amyloid, and although I'm not presenting it here, some other proteins such as neurofibrillary light chain or NFL protein, another marker of neurodegeneration. So in general, if we look at tau in Black individuals, there largely seems to be a split difference such that they either found lower tau in Black individuals or no difference. And same thing in amyloid, where the majority of these studies actually found no difference of amyloid between Black and white individuals, except for one study that did find higher amyloid in Black individuals. So using some data from a recent study, I wanted to look at amyloid in clinically normal older Black adults. And just as a reminder, preclinical AD is the stage in which their cognitive impairment may not be present or apparent to the person or to the clinician. However, proteins can be measured in this preclinical stage, especially amyloid, which is present decades before overt dementia symptoms. And in fact, about 30% of 70-year-olds are actually amyloid positive, and this translates to greater risk of future memory decline and dementia, but it's unknown if this translates to non-white populations. So for this, we use data from the anti-amyloid and asymptomatic AD or A4 study, which is a secondary prevention trial in cognitively normal individuals to explore amyloid in a very large data set. So this led to a second paper from my PI at Stanford, Dr. Mormino, where we looked at the effect of white and Black participants on amyloid and whether these effects were influenced by APOE or African ancestry. So first, we wanted to look at if amyloid levels were different in Black and white participants. So here on the y-axis, we have continuous levels of amyloid SUVR, and on the x-axis, we have measures of amyloid in Black and white individuals by genotype, E3E3, E3E4, and E4E4. So just by looking at this, you can see that Black individuals in the A4 study have lower levels of amyloid in the brain. However, this was only significantly different in E4 carriers, suggesting that E4, again, does not confer the same risk for these AD phenotypes in Black participants relative to white participants. I then wanted to look at African ancestry and determine if this was associated with amyloid. So again, I'm bringing back the same figure I showed before, but these are individuals in the A4 study who self-identify as non-Hispanic Black, and their African ancestry exists on this continuum between 30 to 80%. And I just want to make a note that this continuum of African ancestry is present in almost every other cohort that does look at African ancestry, or in every, not almost, in every cohort. On average, people in the United States who identify as Black, their African ancestry does range between 30 and 98%. So we have amyloid SUVR on the y-axis, and we have increasing percentage of African ancestry on the x-axis. And as you can see, as we increase measures of African ancestry, you see that it's correlated with lower amyloid SUVR levels. And this effect of African ancestry on amyloid was actually independent before, so suggesting a potential separate mechanism. Yeah, so even though Black participants were more likely to be diagnosed with Alzheimer's disease, genetic ancestry may influence AD risk factors. However, why might we be seeing these contradictory findings if E4 is the largest genetic risk factor, yet E4 Black individuals have lower amyloid and is conferring the same risk for Alzheimer's disease? We wanted to explore why that might be. So one of these reasons might be selection bias into the A4 study. So around 6,700 people underwent screening and 4,500 completed amyloid PET, but there were about 2,200 people who did not have amyloid PET, which we inferred to be those that failed screening. And you can fail screening for a number of reasons, and that is like CDR greater than 0, mini mental state exam greater than 25, logical memory less than 6 or greater than 18. So indeed, we did see that Black individuals in the A4 study were more likely to screen fail in the study relative to white individuals. And why is that? Because Black individuals were more likely to have too low of cognitive test scores, which again, referring back to not only my Tom 40 findings, but the figure I showed in the introduction, where on average, if we're looking at one time point, Black individuals have a low, on average, a lower score relative to white individuals, while white individuals were more likely to screen fail because of having too high of cognitive test scores. So how can we really address racial bias in current neuropsychological testing if we continue to use these as parameters to screen into a study? So I just want to give a summary of what I've talked about so far before I move on to the next part. And that is APOE4 is associated with cognitive decline in both Black and white individuals, but Tom 40 may aid in better prediction of early cognitive decline in Black individuals. Black individuals also have lower amyloid relative to white, which is driven mainly by APOE4 and high African ancestry. However, Black individuals were more likely to screen fail out of a study for having too low of cognitive test scores. So this leads to a really big question, and that is, who are the Black individuals that we currently have in our studies? If they are performing at a higher level relative to the average Black population, could it be that the Black individuals we actually have in our cohort studies, that they're actually more resilient, to say, for example? So it's going to be hard to identify a true AD risk profile in Black individuals if we continue to use neuropsychological tools that were developed and validated in white individuals. Next, I wanted to talk about looking at computerized cognitive tests. So I am not a clinical neuropsychologist, but I'm trying to pretend to be one these days. And so I've gotten really interested in, again, looking at these neuropsychological, the racial bias that exists in these tests. And so I'm learning about computerized tests. So pen and paper tests, there might be potential bias due to socioeconomic status, educational background, these discrepancies that do exist between Black and white individuals. And computerized testing may reduce these language proficiencies. And on top of that, I know another potential issue with pen and paper tests is that administration and scoring can be different between different groups. And so having a computerized test leads to a more highly standardized output. So there was a test in the A4 study called the Computerized Cognitive Composite test that can detect AD, excuse me, related cognitive impairment and has been associated with AD phenotypes, such as amyloid status. Again, just bringing back this chart to look at different cognitive domains. The C3 battery tests a number of different cognitive domains: episodic memory, working memory, visual, spatial, psychomotor speed. And in general, how this test works is you have an iPad or computer of sorts. And so, for example, if we're looking at the one back test, they just use playing cards for the one back test as opposed to other figures. And playing cards is supposed to be more universal. So this test measures accuracy. So for example, higher is better, or response time, and higher is worse for that. This leads to another study that I'm hoping to get out very quickly. And this is looking at ethnic and racial bias existing for the computerized neurocognitive battery. If what I alluded to in the introduction of this section, focusing on the fact that there might reduce the cultural bias using a computerized test, I would hypothesize that we might see less of an effect in across racial groups with this test. So a lot of this work was led by a PhD student in biostats here at UCSD, Shinran Wang. So we wanted to look at, again, C3 performance across ethnic racial groups. We did control for sex, APOE status, years of age, years of education, and amyloid SUVR. So anything to the left or right of this line, if it does not cross it, is considered significant. So for example, all the tests that we've shown here, behavioral pattern, first letter name and call, one card learning, mainly episodic memory tasks, or working memory tasks, all of the Black individuals still performed at a lower score, still had a lower score relative to white individuals, and same thing with Hispanic individuals as well in the A4 study. We rarely saw sex differences except for in some episodic memory tests. And even education was very moderately associated with this task in these groups. Same thing here, they're on separate graphs because it's an opposite effect. So here, anything to the right suggests that they perform at a lower rate relative to other groups. So again, for example, Black and Hispanic individuals are having a lower score relative to white individuals for these tasks: detection, one back, and identification. So Black and Hispanic participants still performed worse on all the C3 tests relative to white participants. And it appears that racial bias still exists for computerized neurocognitive batteries. However, it's super important as I've grown as an independent ethnic and racial disparities researcher. I am learning this very, very important lesson, and that is we need to stop comparing our data to white individuals. They are not the gold standard, but whiteness continues to be centralized in research, even though my previous work and others show that predictors of AD and cognitive decline differ between Black and white individuals. So that's why it's really important to shift our focus to identifying predictors within the Black community. So I basically reran the same test, but this time I looked at variables within ethnic racial group to correlate. And I figured the easiest way was to kind of do this like color coding block thing. So if a variable was associated with cognitive performance, it's highlighted. So for example, here, education was significantly associated with memory performance in Black individuals, but only age was associated with memory performance in white individuals, as opposed to sex or gender, which was associated with memory performance in both Black and white individuals. So just from taking a big look at the overall color blocks in here, we can see that there are different predictors that are associated with cognitive performance in both Black and white individuals, which again suggests how important it is that we stop comparing, because I think we're losing a lot of work related to what is important within a specific group. So this leads to a big piece in my field as well, in my research that is missing, and that is, focusing on other psychosocial factors that we can use to fully understand risk and resilience. And this is using the Health and Disparities Research Framework from Dr. Carl Hill, who's now at the Alzheimer's Association, but put this out when he was at NIA. And I think it's really important that, a lot of us have been focusing on biological factors and how, they, like proteins and genes, relate to cognitive function or disease status. But it's so important to start to incorporate all of these other factors, environmental, social, cultural, and behavioral, and how those either interact to promote cognitive function or might have their independent effects. And I want to highlight just a number of different studies. A lot of great work has been coming out recently that it's been focusing on, the other psychosocial factors. And I think it's shining light into the areas that there needs to be some more focus on. And I really want to highlight this paper as well that looks at genetic admixture in the Cuban population as a great example of this. So essentially, this is looking at African admixture in the Cuban population. And similar to what I showed previously, people who identify as Black, which is in these blue dots, you can see their African ancestry really exists on this continuum relative to people who identify as white, which have very low African ancestry. And one of the unique things about this paper is that this Cuban population, there's very few health and social disparities, as the author alludes to. So the potential for social and environmental confounders, it's really limited in this group. So it really allows us to focus on, is there this genetic risk of ancestry? And in fact, whether once you start adjusting by these different variables, there's African ancestry, and these dots represent the effective cognition, cognitive function. You can see that their greater ancestry wasn't associated with worse cognition as a function of African admixture. This leads me back to this slide about why might we be seeing contradictory findings in people who self-identify as Black. I think if you combine the information that I just showed with our knowledge that we've been growing in the field on psychosocial pressure, on psychosocial factors, it shows just how important for people in the United States, structural and institutionalized racism and oppression has on cognitive function later in life. So it's super important that we continue to include in our research these, as it states up there, fundamental factors for cognitive function, and focus on environmental, sociocultural, behavioral, and biological factors. And I want to just also bring to light a lot of great research that's been going on. Some really great papers focusing on, anti-racist behavior, creating research justice, and how important it is that we do this information correctly and not be a health equity tourist. And so with that, I'd just like to say thank you for your time. I finished that a little quickly, but yeah, I've had a lot of help from my UC San Diego group, Stanford group, and a group I started adding is my mental health supporters, because we all know it is hard to work in science, let alone if you're a minoritized individual. And I have a lot of wonderful Black women that scientists that I am able to lean on during my research. So with that, I'll take any questions. You're muted, Talia.
Oh, sorry about that. Thank you so much for that great talk. A very thorough and interesting discussion. We do have one question in the chat. I'll just let everybody know or remind everyone to please put any questions that you may have using the Q&A tool, and then we can answer them live with Dr. Deters. So there is one question here. I believe this was asked earlier in the presentation while you were discussing the research relating to rates of cognitive decline and how it's typically been shown to be variable, but now the impression is that possibly the rate of cognitive decline across racial groups is maybe not different. So this question is asking, is it possible that neuropsych tests used to measure cognitive decline impairment can also be inherently biased towards minority groups?
And I am muted now. Yeah, absolutely. I think that's what a lot of the work goes to show, is that there is the current neuropsychological battery is completely biased towards like minoritized versus white individuals. So I know there are some groups working to create some culturally sensitive tests. And actually, the computerized battery is one of them, supposed to, remove some of these things. So it's interesting that I question myself, I'm relatively new to, neuropsychology work. And yeah, I think it's interesting to see I'm interested to see how the field is going to work towards creating these culturally sensitive tests, and then how can we potentially actually compare, these results between groups. I do think that the current tests we have do measure cognitive function, cognitive ability across all groups. It's just when we use these tests to compare, there's always going to be a disparity. Just like even in the protein analysis that I showed, there's this disparity that's present. So I think that they function within group, but when we start looking across groups or between groups, that's where I think we start to see some issues.
And the basis for some of these comparisons about differences in risk and change are based on those studies.
Yeah.
So the next question, I have several questions popping up now. So this next question is asking, how have you been able to reach the Black community to participate more in research studies? So I'm not sure if this is specifically something that you've done individually, but any thoughts about this? Go ahead.
Yeah, that's a great question. I have not done that individually. I am hoping when as I transition to a faculty job, whenever that happens, that's something that I'm going to work towards. I have really great mentors who have done a lot of work and have experience. A special shout out for Dr. Lisa Barnes and Dr. Jennifer Manley, who have taken me under their wing a lot. And I look forward to learning a lot from them about how to engage the community in the appropriate way. And again, just going back to the topic on health equity tourists, there's definitely a right way and not so right way, to work with minoritized groups. And it's super important that, we learn from people who have these experiences before we jump on there.
Yeah, I would agree that there's lots of examples of people doing community-based or real enriching, I guess, community-based research where they're, directly involved with community and therefore have better access to some of these populations. Mm-hmm. Okay, we have another question. I see a question that actually I think went to the chat, but I'll try to stay focused on the Q&A here first. So this question stating, hi, I had a question related to amyloid levels in Black versus white individuals. You showed reduced amyloid PIB-PET in Blacks, I think, using A4 data. As mentioned, this may be a resilient Black cohort. Has this been replicated in other samples, and how does tau accumulation differ between Blacks and whites?
That's a good question. No, this has not been replicated in other studies. There was another PET study that actually showed different results than us, and that was from Gozman, I think it's Rebecca Gozman. And they actually showed higher amyloid levels in the Black participants relative to my findings where it was lower. And then other groups actually found no difference in amyloid levels. So the question disappeared, so I forgot what one of the, oh wait, no, it's in the answer, so I can read it.
Yes.
Okay, yeah, I was just, so no to the replication part. And then regarding to tau, I'm uncertain. I haven't studied tau extensively at all in the Black community, so I don't quite know how accumulation might differ between Black and white individuals. But I think one of the points that I tried to make by showing the comorbidity chart from Dr. Lisa Barnes and other people have also looked at postmortem results is that it's not just amyloid and tau. Even in the white individuals, there was a lot of other potential comorbidities. Amyloid and tau have been the focus of the Alzheimer's disease world because of, I think because they were the earliest found proteins. But now as we have these other techniques to actually identify other proteinopathies, I just think that there might be other factors that contribute to cognitive decline or impairment or dementia in Black individuals relative to white individuals. And then, yeah, I think that was it.
Answer both questions. Okay, so this question has now been posted in the Q&A. So given that there are no truly culture-free tests, which features of new neuropsychological test construction will be most important to prioritize to improve our neuropsychological batteries for cross-cultural comparisons?
Yeah, so I think having culturally competent researchers in the field is super important. Again, I just keep going back to the health equity tours thing because I think it's a really important thing to think of. And the paper is by Owlett, I believe was the name, that discusses a lot about this based on their experience. But I think having cultural competence is extremely important. Understanding the culture is going to really, really help with trying to remove some bias as well. But other than that, again, I am not a trained neuropsychologist, so I'm learning these things right now. I'm super interested in it. But I would definitely, that's the number one thing I can think of right now.
Having some forethought before measures are constructed,
Yeah.
Absolutely. Okay, so I don't believe I see any more questions in the chat here. I'll make sure to double check. But I did have other kind of questions related to some of your work, general ideas, and also maybe some discussions that we've also had is in talking about evaluating either differences or disparities across different variables when assessing Alzheimer's disease risk or cognition, is the way that we measure race, or the way that we use race as a variable in this kind of categorical variable. Asking individuals to identify as Black or white or Hispanic, it really does take out a lot of the variability and cultural experiences within these groups. And how do you feel like that is affecting the research that we have on race and thoughts about going forward? And how do you feel like admixture might, research on admixture might help us out or contribute to how we can ask those questions in more nuanced ways?
So I'm sorry. So you're asking
me questions there.
Yeah.
So how do you feel like the way that we ask about race, or the way we ask individuals to identify based on race in ways like, using, for example, population of people who identify as Black in the United States is a very mixed group of people. The population of people who identify as Latinx in the United States is a very mixed group of people. And is that contributing, or how might that be affecting some of the work that we see?
Yeah, thank you, Talia. Absolutely. So I think self-identification is one thing, but I think this is where the paying attention to the disparities framework, where we look at environmental, social, is really going to come into play, because I believe that a lot of these disparities, and I think that a large group of us is due to the systemic racism and oppression and that is currently has been going on, that has forced people into a different group, I should say, that don't have, access to healthcare, health equity. So I think, it's still helpful. I still think that, there's information, but if anything, it's just really like a really, really, really poor proxy to socioeconomic status. So yeah, And then as far as admixture, that's something I actually hope to pursue in my future research. So again, like a lot of my interest has been driven from myself because I'm mixed and Black and white. And I think having different cultural pressures from different groups, I'm unclear or unsure about how that actually impacts, a person's later life cognitive function. It's a different type of bias, a different type of discrimination that you get from, say, for example, somebody is mixed like myself, identifies as Black, and you have discrimination, from both sides. How might that actually contribute to later life cognitive function? So yeah, that's kind of a way that I'm going to look at admixture, I think, in my future research is really focusing on like psychosocial factors across the lifespan for admixed individuals.
Thanks. Thank you. I don't see any other questions, so I'll just kind of keep bugging you with my own. So this is kind of related to what you were just mentioning, and also related to your final points in your presentation about the importance of understanding these questions or asking these questions with this NIA health disparities research framework. And I know there was a recent paper that you and I had discussed talking about how we define or how we use race in research studies, and how it's really, like you said, a proxy for all these other things. And that in reality, when we're looking at disparities, race as a variable is maybe not necessarily evaluating any type of biological process, but it's actually more so evaluating racism or structural racism and differences in life experiences, and how that should probably be centered more in the research that we have going forward. So I don't know if I have a specific question, more of just a comment and open discussion for your thoughts.
Yeah. I mean, sorry, I hate to reference social media, but the science Twitter really changes my perspective on a lot of things. And I actually saw somebody who had made this quote about, I didn't realize how big of an impact it had on me. And I don't even know if I liked it, because but to go back and look, but they were like, in 2022, let's remove race as a variable and start thinking of it as racism. Once you start to think about things that way, then, when we do our research comparing, where people are trying to compare differences between white and Black, it's never Black. Black is not the problem. It's this racism and systemic oppression and everything else that I've already said a few times in this presentation, that is really what's causing the disparity. It's not being Black, it's not being Hispanic, Latinx, it's not being Asian. So that really is, I think, like a life-changing view that I've had recently, and just making sure that when I'm doing my research, that I am taking the time to try to make sure that there are other factors that I could use in my analysis to determine, what is truly, if I see an effect, what is truly leading to this disparity between the groups, because it's not just being Black, it's not just being Asian, So.
Right. Well, thank you so much. I don't know if I've missed any other. Oh, I do have another question. Here we go.
Mm-hmm.
Okay, so how should we ask participants about their racial and ethnic identity?
Yeah, I think it's, how do you identify? I think it's as simple as that. I think when there's a lot of boxes to be, and offering people the potential to select a lot of different boxes. I think an issue, again, I keep going back to myself because I'm mixed. I think an issue that I see that isn't reported a lot is, we look, people that come into a study, and they're asked, how do they identify? And then they might mark a box as Black. They don't have an opportunity to mark more than one. Or if they do have an opportunity to mark more than one, in the cohorts I've studied, there isn't a section for race or ethnicity that allows admixture. It's always just one of those groups. Mm-hmm. So I think it's going to be really important. And one thing I've noticed in like working with the NAC data set, or the National Alzheimer's Disease Coordinating Center data sets, is that they do have a columns that have like a first, second, third, and I think, those are to identify people who identify as ethnic. So again, for myself, because I am Black and white, I do have lighter skin, which means I might have a different social experience than somebody who has darker skin. So, all of these things have such an impact on, our overall health. And I think it's really important to just ask, how do you identify? And if it's more than one, we need to start having a different section that looks at this, admixture in itself. And again, it's going to be a smaller group, but if we keep working toward that, hopefully we'll end up, with growing numbers of participants.
I apologize for my honking if you heard that. No, I was thinking related to that question is, there's asking about people's, self-identified race, but there's other aspects of their cultural identity that are related to race that might be important. And I guess we could think about how you could use those in research, but ultimately having a discussion recently about this similar question, in terms of using someone who identifies as Black as an example. Are they an immigrant from another country? They're, people who have Caribbean origin. How long have they been in this country? What regional differences? All those are different things that might play different or represent, sociocultural differences that might contribute to life experiences in important ways. So yeah, those are just other things, besides just the question of identifying your race or ethnicity that are relevant and might play a role in some of the variability that we see, particularly as it relates to this kind of sociocultural influences. So
absolutely.
Yeah. So we are at 12:55. I don't know if we have any other questions or comments from any of our panelists, but if not, I really, really appreciate your discussion, your talk, and showing your research, and also your thoughts for future research and how we move forward addressing these important issues and questions. Don't want to kick people off, but I don't know if I have that power, but we're coming to the end here, but I just really wanted to say thank you.
Thank you.
For allowing me to introduce you, and it's been fun chatting.
Thank you so much. I really appreciate being given the opportunity to talk about my research.
Thank you.
Thank you very much. And thank you also, Talia. Thank you. Bye.